So, hello everyone, welcome to my channel SS Medico. Today we are going to discuss an important topic of cancer, that is conversion of proto-oncogene to oncogene. So, first of all, what is proto-oncogene? You have to remember that these are normal genes which presence in our body, which are responsible for production of various important things which are important for our growth, that is like various transcription factors like growth factors, receptors for growth factor, and proteins that regulate gene expression. So proto-oncogenes are the normal genes in cell responsible for production of various transcription factor, growth factor, their receptor, and proteins that regulate gene expression. So proto-oncogenes are normal structures. Now what happens, due to some process like some point mutation, translocation, gene amplification, or some insertion deletion, these proto-oncogenes are converted into oncogenes. Now these oncogenes are genes capable of causing cancer, which leading to mutated proteins, leading to uncontrolled cell division and leading to cancer. So now we are going to discuss the process of conversion of those proto-oncogene to oncogene. So this is done by 5 processes. So the first process, it is the promoter insertion, where in the promoter region there is a mutation. So the mutated promoter cause an overproduction of a stimulatory protein causing cell proliferation. For example, suppose this is a normal chromosome, normal chicken chromosome, which have a inactive MYC gene. So this is a normal chromosome containing a normal MYC gene. What happen, suppose there is a virus infection like the avian leukemia virus, bird leukemia, avian leukemia virus in the promoter region. So promoter means mainly the upstream region. So this avian leukemia virus, they insert their gene, insert upstream to the inactive MYC gene. So this is the inactive MYC gene, and this virus, it is inserted into the upstream region, so the promoter region. So and also some LTR region are seen. LTR means some long terminal repeats, which are the strong start signals, long terminal repeats. Now this promoter region is mutated. This will force the cell to constantly transcribe the MYC gene into mRNA and then build proteins non-stop, turning a normal gene to oncogene. So this is how a proto-oncogene converts into oncogene by doing mutation in the promoter region. Enhancer insertion, this is simple. There is a inactive MYC gene, and if the avian leukemia virus, it occurs in the downstream region. Basically in the downstream region, it is enhancer region. So that will lead to uncontrolled protein formation. So promoter insertion, enhancer insertion. Now we have came to an important part, that is the chromosomal translocation. So what is chromosomal translocation? This is the process where a piece of one chromosome splits off to join another. So suppose one chromosome, they have one part that splits from that region and goes to the other chromosome, okay. There is an important example of this chromosomal translocation which occurs in CML, that is chronic myeloid leukemia. It usually occurs in chromosome 9 and chromosome 22. Let's discuss the normal phenomena and what occurs in CML. So suppose this is the normal chromosome 9 and normal chromosome 22. Chromosome have one P arm, one Q arm, one P arm, one Q arm. And you have to remember, in chromosome 9, in the Q arm, and there is a normal gene present, that is the ABL gene. ABL means the full form is Abelson Murin leukemia gene. So ABL gene present in chromosome 9 Q arm normally, and in the chromosome 22 Q arm, there is a normal gene present, that is BCR gene, that is breakpoint cluster region. So you have to remember, chromosome 9 Q arm normally ABL gene present, and chromosome 22 Q arm normally BCR gene present, okay. Acha. Now in the CML, what occur, this T922. What do you mean by this T922? It means that the one part of the 9th chromosome goes into the 22nd chromosome, and that special chromosome is known as Philadelphia chromosome. What happens, the Q chromosome, in the 9th chromosome, this ABL gene is goes into the 22nd chromosome. So now 22nd chromosome has both the ABL gene and also the BCR gene, forming the BCR ABL fusion gene, BCR ABL fusion or hybrid gene, and this chromosome is known as Philadelphia chromosome. Now what will happen, this BCR ABL fusion gene will cause abnormal tyrosine kinase activity. It will cause abnormal tyrosine kinase activity, which will lead to abnormal uncontrolled mitosis, especially in WBC precursors, leading to chronic myeloid leukemia. So this is a very important example of the Philadelphia chromosome, that is here chromosomal translocation. Also this occurs in Burkitt's lymphoma, which is T814. Part of 8th chromosome goes into 14. In CML there is part of 9 grows in 22. Okay, so this is chromosomal translocation. Now remaining two, one is point mutation. Point mutation, we are giving an example. First see what is happening in normal cell and what is happening in cancer cell. So there is a gene, that is the RAS gene, which gives rise to a P21 protein. Now what is the importance of this P21 protein in our body? It acts as a on off switch for our cell growth, on off switch for our cell growth. So whenever it is signaling on signal, it will activate adenylyl cyclase, that will increase cyclic AMP, leading to protein kinase activity. And when the protein kinase activity or growth is not required, it will off the signal and by some GTPase activity. So this is normal mechanism. In cancer, what happened, there is a point mutation at position 12 of P21. Remember, like 1 2 2 1. So there is a point mutation at position 12 of P21. So what happened then, there is a breakage of the off switch. So this off switch is not even functional now. So the protein stays stuck in on position. So what happened, there will be overproduction of cyclic AMP and protein kinase activity, leading to uncontrolled tumor growth. So this is the example of point mutation. Now finally the gene amplification. Suppose this is the one coding region and the oncogene, what happened, there is a multiple copies of this coding region happen. So the oncogene having multiple copies, thus producing larger amounts of the coded protein. So if there is larger amount of coded genes, there will be larger amount of coded protein production. What is the example? We know there is a cancer drug, that is the methotrexate, which inhibit the dihydrofolate reductase enzyme. So for example, in cancer cell, if gene amplification occurs, so there will be hundreds of extra copies of DHFR gene. So there will be very much increased production of dihydrofolate reductase enzyme. So the methotrexate drug will be overwhelmed. So that's how the cancer cells will be resistant to methotrexate treatment. So this is the gene amplification. So these are the 5 important processes: promoter insertion, enhancer insertion, chromosomal translocation, we have seen the Philadelphia chromosome in chronic myeloid leukemia, then the point mutation, and finally the gene amplification. Thank you.